All right, this is a very simple overview of some of the nuclear and cytoplasmic steps that are involved in the expression of a mammalian gene that encodes protein. I know you know them all, but I just want to point out some features. So you know that DNA is transcribed in a collinear fashion to form pre-mRNA that you've heard a lot about already. And then pre-mRNA is processed both co- and post-transcriptionally to generate the mRNA. And again, this is all in the nucleus. The mRNA has a cap at the 5' end, a poly A tail at the 3' end, and if the mRNA underwent splicing, if its pre-mRNA contained introns, which are the black lines here, then it will have lost those introns. Then the mRNA is exported through the gray boxes that represent nuclear pore complexes into the cytoplasm, where translation occurs. And what I'm going to focus on is mRNA decay in the cytoplasm. But before I start talking about that, I want to just mention that transcripts can be degraded in the nucleus. For example, if the pre-mRNA did not obtain a cap co-transcriptionally, then it will not associate with the proteins that bind to the cap, which protect the mRNA and the pre-mRNA, in this case, from decay, so that the uncapped transcript will be degraded by what's called RAT1. This is a nuclear 5' to 3' exonuclease. And as another example, during the process of mRNA 3' end formation, which involves an endonucleolytic cleavage, followed by the addition of poly A to the 5' cleavage product, there will be also a 3' cleavage product that is generated. So what's going to happen with that? Polymerase is continuing to transcribe the DNA, and it turns out the same exonuclease that degraded the uncapped 5' end will degrade the uncapped 5' end of the endonucleolytic cleavage product. That's the 3' cleavage product. Its processivity, its rate of decay, is much faster than the rate at which RNA polymerase II is synthesizing RNA. And so RAT1 will catch up with polymerase and torpedo it off of the chromatin template. And this is a way for cells to prevent transcription into flanking genes, for example, or other regions of the genome that shouldn't be transcribed. All right, you all know what an mRNA generally looks like. We've already talked about the cap and the poly A. There's an open reading frame that is flanked by a 5' translator region and a 3' translator region that begins with the normal translation termination codon. There are exceptions to this structure. So for example, cell cycle regulated histone mRNAs lack a poly A tail, and this becomes important when we think about histone mRNA decay. Also, histone mRNA decays and, for example, C-Jun mRNA, which encodes a proto-oncoprotein, do not derive from splicing. So they will not contain this little line here when they're diagrammed, and that line represents an exon-exon junction. Generally, mRNA stabilizing or destabilizing elements reside in the 3' untranslated region, and we will talk about that. What about mRNA decay? So all cellular mRNAs are eventually degraded. mRNA decay is a highly regulated process controlling gene expression at the post-transcriptional level. And mRNA decay is often coordinately or concomitantly regulated at the transcriptional level. And that makes a lot of sense, because if it's time for the cell to eliminate the production of a particular protein, and it does so by degrading the mRNA that encodes that protein, it behooves the cell to also stop synthesizing the mRNA. mRNA half-life is controlled by specific interactions between cis-acting mRNA elements and the proteins that bind these elements. Oh, I'd like to just remind you, half-life means the time that it takes to degrade half of existing mRNA molecules. Half-life experimentally needs to be measured after the experimentalist has blocked ongoing transcription, ideally specifically of the gene that encodes that mRNA. And then it's the amount of time that it takes to degrade half of the molecules from that point on. The cis-acting sequences that regulate mRNA stability can be general. We've already talked about the cap-binding proteins that protect the 5' end of an mRNA from RAT1, exonuclease in the nucleus, and the corresponding exonuclease in the cytoplasm called XRN1. The poly-A binding protein protects the 3' end of the mRNA from deadenylation. So these are general stabilizing factors. But there are also specific factors, such as factors that bind to AU-rich elements, which are invariably in mRNA 3' untranslated regions, and they are generally destabilizing by virtue of the proteins that they associate with. And you can imagine the process of mRNA translation can influence the rate at which an mRNA is degraded. For example, translocating ribosomes can displace a protein that binds to the open reading frame of an mRNA and stabilizes the mRNA. And we'll provide some examples of that. Eukaryotic mRNA half-lives can vary from minutes to hours. Generally, the faster a cell divides, the shorter-lived its mRNAs. So bacterial mRNAs have much shorter half-lives than mammalian cell mRNAs. And I've given a few examples here. Beta-globin mRNA, it's usually present in erythroid cells, has a half-life of about 17 hours, whereas C-Jun mRNA, which encodes proto-oncoprotein, has a very short half-life of 15 to 20 minutes. And in fact, one way to transform a cell is to increase the half-life of C-Jun mRNA, and the viral Jun mRNA has a much longer lived half-life than its cellular counterpart. Changes in mRNA decay allow for the fine-tuning of gene expression, and they can occur in response to developmental or environmental stimuli such as nutrients or cytokines, hormones, temperature shifts, as well as environmental stressors. As one example, the mammary gland mRNA for casein, which is a milk protein, is increased from 5.4 hours to 92 hours when mammary tissue is exposed to the peptide hormone prolactin. And concomitantly with this, casein gene transcription levels are upregulated 2 to 4-fold. As another example, the cell cycle regulated histone mRNAs are degraded at the end of S phase of the cell cycle when the cell doesn't need to make histones anymore. There's no more DNA replication. And therefore, histone mRNA is degraded with a half-life of 15 to 20 minutes, whereas at other points in the cell cycle, it has a much longer half-life. And with that decrease in mRNA half-life at the end of S phase, histone gene transcription is also downregulated. This illustrates that a very small change in mRNA half-life can manifest as a rather large change in mRNA abundance. And the example here is of an mRNA that has a half-life of 40 minutes compared to an mRNA that has a half-life of 10 minutes. This is providing that the transcription of the genes from which each mRNA derives is shut off. So there's no confounding further contributions to the mRNA levels in the cell. So if the number of molecules of each mRNA is at the beginning of the transcriptional shutoff 10,000 per cell, you can see that by 120 minutes, there's about 2,500 molecules of this mRNA left, but only something like 2.5 molecules of this. So concomitantly shutting off transcription and destabilizing an mRNA is a very effective way to shut off gene expression. There are 3 major pathways of mRNA decay in mammalian cells and in eukaryotes in general. two are dependent upon deadenylation as the first step. So the poly A tail during the deadenylation process is shortened to an oligo A form. And here are two examples of deadenylases. We'll talk about them in more detail in a moment. Once an oligo A form is created, there are two options. One is that the body of the transcript can be degraded in the 3' to 5' direction by the exosome. And the other is that the 5' end of the mRNA can be decapped by DCP2, which is the decapping activity that's in a complex with a whole bunch of other proteins, including DCP1. Without the cap, then, the uncapped 5' end is available to being degraded by a 5' to 3' exonuclease. We've talked about RAT1 in the nucleus. This is the mostly cytoplasmic equivalent called XRN1. An alternative pathway, which tends to be particular to particular mRNAs, it's not generally applicable to most mRNAs, is the endonuclear pathway, where a 5' and a 3' cleavage product is generated. Here's a list of some endonucleases. And then what happens is, as you can imagine, the 5' product of the endo cleavage is degraded from the 3' end by the exosome, and then the 3' cleavage product from the endonuclear cleavage is susceptible to decay by XRN1. We'll talk about deadenylation first. Anybody have any questions? All right, so there are 3 deadenylases that have been well studied in mammalian cells. And I should mention that these 2 are present in yeast and Drosophila. This is absent from yeast and Drosophila. This complex has a variety of names, one of which is CAF, CCR4, NOT1. There are also POP proteins in this complex. It's much more complicated than this. There are 2 3' to 5' deadenylases, CAF1 and CCR4. They're both active. And notice that this activity is inhibited by poly A binding protein. This is an activity that can be brought to an mRNA. For example, the AU-rich elements, some of them bind tetraproline, just an RNA binding protein, that will recruit this deadenylase via NOT1 as the bridging molecule. And if Marv Wickens were here, he would tell you that mRNAs that are destabilized by PUF proteins undergo deadenylation in a way where the bridge is provided by POP2, a protein of this complex that I haven't shown here. Another deadenylating complex involves these 2 proteins, PAN2 and 3. The active deadenylase is PAN2. PAN3 is thought to be an auxiliary protein. And unlike this complex is stimulated by poly A binding protein. This complex is thought to initially to work on newly synthesized poly A tails and shorten them to a point where mRNAs have a newly synthesized but stable length, at which point this deadenylase, for example, starts to chip away at it. And this is an enzyme activity. These are both members of the RNase D family. They both depend upon magnesium. But this complex will go on and off, and chew a little bit and go off, and chew a little bit and go off, whereas this tends to be much more processive. So this acts early, this acts later, often in a regulated fashion. And then PARN, poly A ribonuclease, is the most active activity in extracts of mammalian cells. But actually these days people are thinking in intact cells, CAF1 is the major deadenylase. This is also stimulated by PAB, and it functions in deadenylating maternal mRNAs of the type that Jim talked about yesterday. So he talked about steps that occurred after maternal mRNA was activated by the process of polyadenylation. And it turns out to keep a maternal mRNA translationally silenced, there's a competition ongoing between GOLD2, which is a poly-A polymerase, and PARN, which is a deadenylase. So I think you all know that cells don't necessarily do things in the way that you would do it if you are going to be the most effective. And in this case, the maternal mRNAs are constantly being polyadenylated by GOLD2, deadenylated by PARN1. Polyadenylated by GOLD2, deadenylated by PARN1, until PARN falls off, GOLD2 wins, the mRNA is adenylated and translationally activated. And for those of you who are wondering, how does adenylation activate translationally an mRNA, poly-A binding protein is known to interact with initiation factors, translation initiation factors that are at the 5' end of an mRNA. EIF4G is a big scaffolding protein that serves as a bridge. It bridges poly-A binding protein to the steady state cap binding protein called eukaryotic initiation factor 4E. All right, we've talked about deadenylases. Let's talk about the exosome. This is a very large protein complex. It has 3' to 5' exonucleolytic activities. And remember, it's going to function after deadenylation to degrade the body of the transcript in the 3' to 5' direction. I don't want you to think that it's only involved in trashing mRNA molecules. In the nucleus, it functions to properly process the extended 3' ends of ribosomal RNA, small nuclear RNAs, small nucleolar RNAs, so that they are mature and stable. And it's also involved in quality control mechanisms in the nucleus that we don't really have time to talk about. This is what we want to talk about. It's involved in degrading proper and translationally defective mRNAs in the cytoplasm. It's been structurally characterized. The core of the exosome consists of 9 subunits, and you can picture it as a trimer it consists of 3 heterodimers, one of which is here, one of which is here, and one of which is here. And on top of that donut-shaped base are 3 proteins that I've put in boxes here that form the cap. So looking at it this way, it looks like a donut with a hole in it, and this is where the RNA threads through. 2 to 9 nucleotides of unstructured RNA can thread through this. Here's a side view where the cap is shown here, and this is the base of the donut. None of these, even though they evolved, or at least look like they could have 3' to 5' exonucleotic activity, none of these have any activity. Their active sites are mutated. These 2 additional proteins associate with the core of the exosome, and they can associate at the same time. They're the ones that have exonucleotic activity, and they form 5' monophosphates by a hydrolytic reaction. This is mostly nuclear. This is mostly cytoplasmic. In yeast, either one can be deleted and the cells don't die. They're debilitated, but they don't die. If both are deleted, it's toxic. So there must be, at some level, some functional redundancy. How does the exosome get recruited to an mRNA? And here are 3 possible scenarios, and there's many more. I'm just providing you with some examples. This could be an oligoadenylated mRNA that has already gone deadenylation. And remember that the exosome can thread an unstructured 2 to 9 nucleotide piece, so it will do that. To get any further, it associates with this complex of proteins, SCI2 of which is a helicase, and that is thought to be required for the exosome to make some progress into the transcribed portion of the message, which often can have structures that would impede it otherwise, or proteins in the way. And the intermediary molecule is SCI7. We've talked about ARE-mediated decay. So this is an AU-rich element in the 3' UTR of a labile mRNA that recruits an RNA binding protein like tris-tetraproline, which, remember, can recruit not one of the CCR4 CAF1 deadenylating complex. Well, after that, it can recruit the exosome. So the destabilizing element first recruits the deadenylase, then recruits the exosome. And there's another way for exosomes to join an mRNA. There's a process called non-stop decay. So what happens if the cell makes mistakes all the time? Polyadenylation occurs upstream of the normal spot, right in the middle of the reading frame, let's say. Ribosomes are going to run to the end of the RNA, and then what? There's no termination codon. This is toxic. You have to recycle ribosomes in order to stay viable. And the way that the cell does this is to recruit the exosome again by SCI7. SCI7 is a molecular mimic of eukaryotic release factor 1, which is a translation termination factor. And so there's an empty A site there on the ribosome, and this will recognize that with the exosome, and the RNA will be degraded. The ribosomes will be recycled for further translation initiation events. I've just told you that these two activities are responsible for the 3' to 5' exonuclease function of the exosome. And I want to mention that RP44 has an endonuclear activity in addition to its exonuclear activity. And that was discovered because there is a pin domain. Nobody really understands the significance of the endonuclear activity. Remember, I told you that you could delete RP44, and as long as you had RP6, you were okay. I mean, the yeast weren't growing extremely well, but they're viable. So this must mean the endonuclear activity is not essential. Quite frankly, nobody really understands how it contributes to mRNA decay. And we're going to talk about SMUG6 when we talk about NMD next time, tomorrow. Also has a pin domain, also is an endonuclease. All right, we've done deadenylation, then we've done decay of the body of the transcript by the exosome. After deadenylation to an oligo A form, there can also be decapping. And decapping of an oligo A form is accomplished by DCP2. And as I said, it's an complex with many other proteins. I think I show some of them on the next slide, including DCP1. The decapping activity requires a long RNA, and it will generate 7-methyl-GPP and a 5' monophosphate end, which is then a good substrate for XRN1, the 5' to 3' exonuclease. There's also a scavenger capping enzyme. So when you think about it, if the exosome is going to be degrading the body of the transcript this way, remember it can only take in about 2 to 9 nucleotides and then it stops. So what happens to this? We need to get rid of this. And the scavenger decapping enzyme does that. It will use this substrate that's left over from exonuclear decay and generate a 5' diphosphate end, which is an okay substrate for XRN1. Decay of mRNAs occurs on polysomes. I'm not going to talk about P-bodies, but I'm happy to answer any questions if you'd like. And as we already talked about, poly-A tails are deadenylated to an oligo A form. Oligoadenylated RNA is favored for binding by PAT1 and LSM17, which is a complex of proteins that are like the SM proteins that are associated with the SNRPs. And they nucleate XRN1 and the activities that I've been talking to you about that facilitate decapping, including the decapping activities. There's also recruitment of the exosome to the oligo A form with SCI7 and this helicase. So mRNA generally occurs, is degraded on polysomes. All right, how do mRNA-specific binding proteins influence half-life? I think I've already told you that generally cis-acting elements that regulate mRNA half-life are in the 3' UTRs. If they're destabilizing, they often target the mRNA for decay from the 3' end. If they are stabilizing, they often protect the mRNA from endonucleolytic cleavage. Here is an example of how people learned about a stabilizing element that's present in the 3' untranslated region of alpha-globin mRNA and how translation can influence mRNA half-life. A long time ago, there was a hemoglobinopathy called hemoglobin constant spring. It's a hemolytic anemia. And the mutation turned out to be a mutation in the termination codon. So that translation didn't terminate normally, but went into the 3' untranslated region. This destabilized alpha-globin mRNA. It turns out it destabilized alpha-globin mRNA because there is a C-rich element here that recruits alpha-CP. It's a complex of proteins, one of which binds C-rich regions. And when ribosomes displaced this complex, the mRNA was destabilized by two mechanisms. There was now accessibility to an endonuclease that cleaved in the C-rich region, and that's because without this complex, poly-A binding protein can't interact to stabilize this protein from binding to the C-rich element. And also without the complex being bound, the poly-A itself was accessible to being degraded by the exosome, by deadenylase, and then the body by the exosome. So there's an example of a stabilizing element in the 3' UTR that, if removed by ribosomes that translate into the 3' UTR, destabilize the mRNA. Another example of a destabilizing element in the 3' UTR that's been very well studied and does not appear to depend upon mRNA translation is the AU-rich element that typifies a lot of cytokine, lymphokine, and other type of growth factor mRNAs. This was discovered a long time ago, actually in 1986, in a Cell paper that has been very highly cited. But the bottom line is, in that paper, beta globin, remember beta globin mRNA has a long half-life in erythroid cells and also non-erythroid cells. It has about a 17-hour half-life in non-erythroid cells. They say greater than 10 here. Can be destabilized if in the gene there is inserted now it would be a T-rich element from the 3' UTR of granulocyte monocyte colony stimulating factor gene. So the mRNA can be destabilized when the wild type sequence is inserted. At the RNA level, it will be AU-rich, why it's called an ARE. And if a corresponding sequence were inserted that had been mutated, so the AU richness was peppered with G's and C's, then the mRNA half-life wasn't affected. The ARE could not confer lability. So here's how the experiment was done. Transient transfections were performed using NIH3T3 cells. The authors used a rabbit beta globin gene. Here are the two introns. They're expressing the rabbit globin gene from the rabbit globin promoter, which can be promiscuously expressed in non-erythroid cells using the polyoma enhancer. And into the 3' untranslated region of the gene was inserted the AU-rich element from mouse. This is murine mouse, granulocyte monocyte colony stimulating factor, ARE, or a mutated version. That shows how the mouse and the human are very similar. Conservation, I think you all know, in 3' UTRs is a good indicator of functionality, conservation among species. Here is the mutated version that's GC-rich. This is a northern blot. So when the wild type rabbit beta globin gene is introduced, the mRNA that derives from it is stable in the sense you can see a band. With the ARE inserted into the 3' UTR, there's very little detectable steady-state mRNA. And with the mutated ARE that is GC-rich, the beta globin mRNA is now evident again. So this is not a half-life study. This is a steady-state analysis. One cannot make any conclusions about mRNA stability from this. We can also only say that the mRNA levels change. But if one were to measure pre-mRNA levels and control for changes in transcription that could occur, which it turns out they don't occur in these experiments, but they didn't know that back then one could infer this is an effect on half-life, which it is. Any questions? And you know what? Did I say it's a northern blot? It's an S1 nuclease transcript mapping, another way to measure steady-state RNA. Yes?
How do you stop transcription?
How do you stop transcription? That's an excellent question. So the way to do it in a sloppy way that will not get you into a nice journal is to use actinomycin D, for example. So the way one wants to measure half-life is not by inhibiting global cellular transcription, because that can have secondary effects. And I'll show you in one of the next slides that use of the CFOS promoter is quite a nice way, because it's transiently inducible when serum is deprived from cells, provided the cells upon serum deprivation go into G0. And then when serum is added back, there's a transient burst in transcription, which is over by 30 minutes. And during that time, then, one can look at half-life. Very nice. And there are the TET inducible promoters now that one can get commercially. That's a very important question, because a lot of people want to measure half-life in a way that is not a good way to do it. How am I doing on time? I'm almost done. Is that right? I almost have to finish.
I forgot when you start.
35. All right. So let me just, I can do it. Let me just summarize, because you're going to be getting these slides as handouts if you're desperately interested to know exactly what was on them. Iron is an important nutrient in cells. It's present in heme, but it's also toxic. So the cells have to regulate their intracellular iron concentration. One of the ways they do that is by regulating the level of transferrin receptor mRNA. Transferrin receptors are on the cell surface and take iron into cells. So the bottom line is, and there's also a way to store iron in cells using ferritin mRNA, and its translation is regulated by intracellular iron concentrations. But here's the bottom line, since I'm running out of time. When intracellular iron levels are low, it is important to have high levels of transferrin receptor so that more iron can be brought into the cell. And the way that the cell does that is to sense the iron concentration through sulfhydryl moieties, and that allows the iron response element in the 3' untranslated region of this mRNA to associate with the iron response element binding protein. And it does so in a bunch of stem loop structures. This physically occludes an endonuclease from cleaving the mRNA and inactivating it. However, when iron concentrations are high, the opposite occurs. No IRE binding proteins are able to bind, and the endonuclease can destroy the mRNA. So this is a way, remember I talked about conditional ways to regulate mRNA half-life? That's a nice example. I also told you that the translation of an mRNA can influence its half-life, and this is an example I want to provide for that. CFOS mRNA encodes another short-lived mRNA. It has an AU-rich element in the 3' untranslated region, but it also has a destabilizing element in its coding region. So there's two ways by which this mRNA can be degraded. The destabilizing element is called the major coding region determinant. And the authors here, and Ben Shui and his lab, generated two constructs that he transiently introduced into NIH3T3 cells. So here is the FOS induction experiment, the FOS promoter induction experiment. One of these constructs consisted of a beta globin sequence with the coding region determinant in the open reading frame of beta globin, and then more beta globin sequences. And the other was exactly the same, but there was a hairpin structure in the 5' untranslated region. This is a great way to block translation. So someone showed using cyclohexamide, which is not the best way in the world, sorry, but again, because it's a general inhibitor of a whole cellular process. But if you put a hairpin structure in the 5' untranslated region of the mRNA you want to block the translation of, that's going to prevent only the translation of that mRNA. And it does so because the 40S ribosomal subunit, in scanning through the 5' UTR, doesn't have a very good unwinding capability, and it gets stuck. So it can't reach the AUG initiation codon. What the authors showed is, these are driven by the FOS promoter, and as I said, when transiently introduced into cells, and then if the cells are deprived of serum, they'll synchronize in G0. Then serum can be added back to the cells. We usually wait 24 hours, add serum back, and you get a transient burst in transcription which is a maximal at about 30 minutes after serum addition, and then an automatic shut-off of the promoter. You don't have to do anything. It automatically shuts off. And during that shut-off time is when you can measure mRNA half-life. So this is the half-life of, it's not the half-life, but it's the level of the mRNA that doesn't have the hairpin structure. And what the authors are showing is minutes after serum addition to serum-deprived cells. Here is a control for deadenylated RNA. One can take cellular RNA and add to it oligo-DT and RNase H, which cleaves the RNA strand of an RNA-DNA duplex, and get a deadenylated RNA. This is a northern blot. So there's deadenylated BFB mRNA. And look, during the transcriptional shutoff, that mRNA is deadenylated. It's getting smaller and smaller in size. At the same time, its body is being lost so that you cannot see as much mRNA, and at 120 minutes you can't see anything. Whereas when the hairpin is inserted, so translation is blocked, there's no translation through the coding region determinant. That stabilizing protein is not removed. Destabilizing protein is not removed, and you can see that the mRNA is fairly stable. And I won't go through this. It just shows the hairpin blocks translation. And you can also look at this, but they showed that CCR4, which is a member of that first deadenylation complex that I showed you, is the deadenylase involved. And they did that simply by overexpressing CCR4, or a mutant version of CCR4. So here's the normal deadenylation rate, decay rate in the presence of empty vector, but that is slowed with a mutant CCR4, and they want to say it's activated with overexpressing CCR4. It's not affected by PARN or a mutant of PARN, or PAN or a mutant of PAN. And this is just an IP where they show that in fact a protein, UNR, that they've HA tagged, that they know binds to the major coding region determinant, can actually co-IP with CCR4, which they've tagged with a V5 tag. So this is an immunoprecipitation followed by Western blotting. And the last thing I want to talk to you about is histone mRNA decay at the end of S phase, which can be mimicked by adding to cells hydroxyurea. That inhibits DNA synthesis experimentally. Remember how I said the exosome, so there's no deadenylation, there's no poly A. How does the exosome get going on this? There's no unstructured 3' end. What the cell does to degrade histone mRNA at the end of S phase is to add some U's, unstructured U's, and then the exosome can get going. And this is a very nice approach to figure out what is at the 5' and 3' ends of decay intermediates in the cell. And briefly, the mRNA is treated with tobacco acid pyrophosphatase, which removes the cap, and then the RNA is diluted. And then T4 RNA ligase is added, and when you dilute your RNA, you're going to ligate intramolecularly. The probability of the ends of the RNA interacting with one another is much higher than the end of one molecule interacting with the end of another molecule. And then one can add primers after making cDNA and PCR up across the junction, the ligated junction, and determine that in the case of Bill Marsloff's lab, a bunch of T's were added. We're sequencing the DNA at this level, the cDNA. And he figured out which is the terminal U transferase, which are the terminal U transferases that are adding the U's, by using siRNA to downregulate the various cellular tutases, and it turns out they're tutase 1 and 3. So this is a very creative way by which cells can get that exosome going on an RNA that has no unstructured 3' end. And with that, I'm going to stop and take some questions.